everyone, and a very warm welcome to all of you in this session. So today, in this video, we will discuss that how can we analyze the data that we have got from the moisture analyzer, and then how can we carry on to perform the statistical analysis or the statistical interpretation of this data. Now, as we all know, that moisture analyzer is a technique or a method by which we can identify the percentage moisture content of any sample. If you want to know more about this particular technique, you can refer to one of our older videos in the MEI Lab in Raurkela YouTube channel, where you will find that how this technique is performed, what is the principle, and what is its importance in the food and other biotechnological fields. So right now, we will be focusing on the data analysis of the moisture analysis data, and then we will move on that how can we perform the statistical analysis. So here in my screen, I have this particular Excel file where you will have basically three parameters. There is the initial weight of the sample in grams, the final weight of the sample in grams, and the percentage of moisture. Now for the demonstration, we have taken 5 samples, and for each sample we have taken 6 replicates. It is 1, 2, 3, 4, 5 and 6. So here we have 6 replicates. Generally we do triplicates, but I have taken 6 so that we can have more and more accurate data. So what we will do is, we are mainly concerned with this percentage of moisture. This is the parameter which will help us to plot a graph in the Origin software, which I will show you just in the due course of time. So at first, our foremost work will be to calculate the average and the standard deviation from this Excel file. What we will do, we'll simply write equal to, then AVE average. Okay. Now we will select all the replicate values. We will close the bracket and enter. So we have got this average. Now in order to save our time, we will move the cursor like this. A plus sign will come. Left click on your mouse and then simply you drag it along so that it's easily copied. So your average values will be automatically copied. Similarly, what you do in order to calculate the standard deviation, we write equal to STDEV, right? And then what you do is simply you select here, then you press enter. This will come, and then what you do, just drag it along, so you will have your standard deviation values. So in this way, I've calculated for all the A, B, C and D sample, E samples. This is for 5 samples. I've individually calculated the average and the standard deviation. Now, in order to not confuse yourself and in order to have a compact tabular data for your graph, what you will do is, we will have this type of table where we will have samples in one column, the average values and their corresponding standard deviation values. So what we will do, in order to copy this value, so this is the percentage moisture value that we copied, right, for other samples. So what we'll do is, we will go to this cell, okay, we will do Control C, let us copy this cell, and then what we will do, we will click on this cell, left click on this cell and simply right click. We will paste values only. Why? If we paste any other thing, then some other value will come, this is undesirable. Okay. Similarly, what you do for the standard deviation, we click on this cell, we do Control C, and we will again click on this cell, left click, and then we will do right click and then Control V. So we will have the average and the standard deviation values in a compact tabular form. Now in this Excel sheet only, for our convenience, we will have a table for our statistical analysis in the SPSS software, which I'll show you again in the due course of time. Now, this is a very important point to note. Whenever we are performing statistical analysis in SPSS software for any kind of data that you want to do, SPSS software does not take any alphabet or letter. It only accepts numbers, right? So suppose if we are considering A, B, C, D and E, we can designate A as 0. So what I've done is, Here we will have to take all the replicate values, that is these replicate values. So the statistical analysis that we are doing here is basically the replicate values of the percentage of moisture. We will not consider the initial weight and the final weight. Why? Because the graph will be plotted against the percentage of moisture itself. So we will consider not this average value, not the standard value, but the original replicate data values of the sample A. And for our convenience, as it doesn't accept any letter, so we have designated sample A as 0. So you have to designate each and every value as a part of A. For that we have done 0, 0, 0, 0, 0 and 0, right? So we will take only the raw data replicates of the sample A. So this is the sample which have for our convenience and to comply to the rules of SPSS, we have to designate as 0 basically here, right? And then we will just copy and paste here the values that we have taken, that is the raw data replicate values. Similarly, for B we have copied, you see, B we have designated as 1, we have copied here. For C we have similarly copied. For D we have done 3, and for E we have done 4. 4 means it's E, 4 for species, and this basically is the percentage moisture data, the replicate values that we have done after that experiment. Okay, so this is the overall Excel file that we have to make before we start any kind of graphical interpretation and statistical interpretation. So right now what we will do is we will go to this origin software. This is our origin software basically. So we'll have a workbook like this, like we have to maximize. So what we will do, we will simply do like samples, average and standard deviation. We will copy this particular folder, this particular table, and then what we will do is we will go to the origin software, or else what we can do for a convenience, we can just simply copy this because we already know. So we will copy and then Control V, it will be pasted. Okay. So the next step will be, as this column represents the standard deviation, now always in order to represent the standard deviation, you have to represent it as error bar. So where we will go, just simply select this particular column and just click here. This is your set as Y error bars. Okay, understood? So we will click on here and then we will select all of these columns and simply we will go to the column graph. So this is your column graph that we generated. Okay. Now first of all what we will do is we will delete this part. Okay. Now we have to make some changes in order to represent it in much better way. What we can do, we can change the labels here. Here will be always your percentage moisture content. Percentage moisture I am writing for our demonstration. This is your, like, you can increase the font size as I have already shown in the previous video. Then you can just simply what you do is you go to labels of the x-axis, double click on it, and then what you do, you can write here samples. As per your convenience, you can write any name, anything whatsoever. And what you can do is you can increase the font, just single click on it, single left click, and then you can increase the font to 24, and you can change the scales also. Now how we can change the scale? Let us change the scale. Now here we have the increment is 20. Let us say from 0 to 55. Let us say from 0 to 70. Let's say, and the increment will be by 10. Increment means like 0, 10, 20, which means the difference between the two values in the y-axis is 10. Difference is 10 basically. So this is your data. So you can basically shorten the graph. You can lengthen the graph as per your choice. Like if I want to like shorten my graph, then I can what I can do is 60 will be enough for me. Okay, so this is my data. Now these are my error bars. So in order to increase the fonts of the error bars, just single left click on it, and then in this you can increase it to 1.5, and then you can make this bold also. Like you can make this bold. Okay, you can make this is also as bold depending on your choice. So this is your 24 bold size, and you can also make it bold, like let me give 22 and bold. So this is your graph that you have done. So your first and primary step will be to plot the moisture graph that is coming from the different samples. So after we tabulate the entire data, we will plot the graph in the origin software. But that's not the end, because we have to again perform a statistical analysis, right? So in order to perform the statistical analysis of this moisture data, we will simply go to another software which is called the SPSS software. So this is our SPSS software. Now before I start my uh, analysis, I just need to tell you that why we do the statistical analysis, because statistical analysis is done in order to discern the patterns and trends, which means that we are collecting large amounts of data from different samples, right? So in order to understand the trends and the relationships between the different samples, like how the samples are varying, and also whether the data that I have collected is right or wrong, whether the data is really accurate or not accurate. So in order to understand the accuracy and the validity of the data, we always perform statistical analysis. Okay, so right now we will go to our Excel file and we will simply copy these values. That is the table that we had made for the statistical analysis. Okay, so we will go to this SPSS software. We will click a single click on here, Control V. We will copy this. Okay, so now we will select these two columns. What we will do is we will go to Analyze. We will go to Compare Means, and we will go to One Way ANOVA. Now this is an important portion. The variable 2 is a value, right? Because this is a variable 2, as you can see from here, which means these will vary. But this is not varying, right? These are basically the sample names that we have designated as numbers. So they will be always in factor, and this variable 2, that is the values, that is the percentage moisture content values, will always be in our dependent list. So variable 2 will always be on our dependent list, and variable 1 will be factor. Okay, now we will do a post hoc test. We will click on post hoc, click on to key, right, and then we will continue, click on OK. So we will have the post hoc tests where they will be analyzing the, or that is they will be basically comparing between the different sample means, which I'll explain to you a little bit later after we do the analysis. So this is our basically the table that we are concerned about. So what we will do is we will simply copy this table. Okay, copy this table, and in another Excel sheet, like in another file, in the same file, we can just paste it here. That will be helpful for us. So now we already know that this is our table, right? So what we will do, these are actually what? These are the means of those samples, right? So in order to have the statistical analysis, we will consider this group as A and this group as B. Okay. Now, first of all, before I go to assign the statistical analysis symbols in the graph, I need to explain to you that why we need one-way ANOVA and the post-hoc Tukey test. Basically, one-way ANOVA indicates, or one-way ANOVA is used to signify or to understand the significant difference among the means of the three or more groups. Like suppose if we have three or more groups of samples, then we will compare those means of the samples, and then we will analyze that whether they are having any significant difference or not. Okay, in case of one way ANOVA. But why we have done here post hoc Tukey test? is because one-way ANOVA will never be able to tell us that which groups differ. That is, in what group is the data varying? That is, which groups have a significant difference, which groups do not have a significant difference. So in order to understand the groups of the data that are having the significant differences in the statistical interpretation, we always use a post hoc cookie test, which means after we have done the one-way ANOVA, which means one-way analysis of variances, where we basically compare the means of the sample populations and try to find out that whether these means have a significant difference or not. But one-way ANOVA will never be able to identify between these means that which group of the means have significant differences. In order to pinpoint or identify the significant differences between the groups of the means of the samples, we must perform a post hoc Tukey test. And post hoc Tukey test is very good because it also eliminates type 1 error, which means if you are considering something as significant, it is actually not significant. Which means if you are considering significant differences by mistake, by chance, then it's called a type 1 error, which is completely minimized when we are performing a post hoc Tukey test. So after the post hoc Tukey test, we will get this table in SPSS where what we have to do is we will assign this in our mind as A, this group, and this group of means as B. Okay, so first of all, 4 means what? It's E. So 4 means it's E. So we will correspond this particular table with our graph in the origin. Okay, so now as you can see from the table that we have got in the SPSS software. Now this 4, as we all know, it represents the E sample. So we have divided these means into 2 groups. One is A group, this is the A group, and this is the B group. So what we will do is we will directly go to the graph in the origin. As we all know that 4 corresponds to E and the mean of the E sample corresponds to the A group. So what we will do, we will go to this origin software and then we will simply hover around this text tool, left click on it, then again a left click, and we will write here A in brackets. Why? Because the mean of this sample E after analyzing all the replicates is assigned to the group A. In our minds, we have already done that. Next, what we will do, we will again go to the Excel file and then we will go for the next sample. The next is 0. 0 means it's the A sample. Now, as you can see from this particular table, that the means of this particular sample is present in both the groups, that is A as well as B. So what we will do, we will simply hover around the origin graph. Again, we will go to the text tool, left click on it. We will go to the particular location just above the error bar, left click again, and then in brackets we will write AB. So here it is A, right? It's not in D. So what we will do, we will go to the A and then we write A. Okay. Then we will again go to the Excel file. Next is 2. 2 means it's C. 0, there is A, B, C and D, right? 2 is, so we have to understand that in 2 it's A and B. So just we will calculate is 0, 1, 2. So in 2 also it's C. That means we will simply control C and then control V. Okay. And we should not confuse the significant like the statistical notations. Otherwise, your entire analysis will go wrong. So here 2 means it's C. So in C we have A and B. That is the mean of the C sample is correspond to both the groups A and B. It's falling under both the groups A and B. So we will do AB. Again we go to the 1. 1 means what? If we go to the graph, 0 and 1. 1 means B, right? So in B also we see it's A and B. So what we can do, to save our time, we'll simply click on this, Control C, and then we will do Control V to copy and paste it, and then we will drag it along just above the error bar. So we must remember that whatever statistical notations we are giving, it must be above the error bar in brackets. Okay, and then again we'll go to the Excel file. The last is 3. 3 means it's D. So D sample, the means of the D sample after one-way ANOVA and post hoc analysis is falling in the group 2. So what we will do is, we will again go to the origin graph. Again we will go to the text tool, left click on it, just above the error bar, we will go again a left click, and then we will simply type bracket. So so we have done giving our statistical notations. Now from this graph, how can we interpret the data? Like when you will do an experiment in moisture, and when you want to interpret the graph, how will you be able to interpret the graph in a statistical manner, and what is the importance of these statistical notations? I'll explain now itself. Now as you can see from this entire graph, the sample D has the highest moisture content, right? It's evident from this graph. And sample E has the lowest moisture content. Okay, and if we compare among this A, B and C, we can see that although like B has little bit higher moisture content, percentage moisture content as compared to A and C, and with respect to the other samples also, but these three samples, that is A, B and C, they are statistically similar. Now why? Because the means of these particular groups, that is the A, B and C samples, the means of these particular samples, they are lying in the same groups. That is AB, AB and AB. Now AB means what? This is my A, this is my B. So we must keep in our minds that the first column corresponds to the A that we have given in the graph. Okay, and then the second column corresponds to the B that we are given from the graph. Okay, so these three samples, that is A, B, they have statistically similar data. That is, they are not significantly different, which means in statistical terms the p value is greater than 0.05. Now if we consider the D sample and the E sample, we can see from here that this D sample has the highest moisture content among all the samples, right? And from the E sample also we can see that it has the lowest moisture content. Now if we can see very minutely, this D sample and E sample, they are statistically significant, which means they are significantly different data. Why? Because the means of this D sample and the means of this A sample, they are lying in the two different groups. So whenever they are lying in two different groups, it clearly signifies that they are statistically different. But now how can I correlate between the other samples as well? So if you want to write in a holistic approach any kind of result, if you want to interpret, you have to interpret in terms of all the samples. Therefore we can say in this way that the sample D has the highest percentage moisture content among all the samples, and it is statistically similar to these C, B and A. Why? Because this B letter is there. Now this it means that the mean of this D group of sample, that is the groups of means of this D sample, and the groups of means of the C, B and A samples, they are lying in the same group, which is the B. As I have shown you already, that B group means the second column. That's why this D sample is statistically similar with respect to the C, B and A sample, but it is significantly different, or it is statistically significant with respect to the E sample. Why? Because the groups of means of this D sample and the groups of means of this E sample, they are lying in the two different groups, which means that in statistical terms the p value is less than 0.05. Therefore, we can say they are statistically significant or significantly different in terms of statistical terms. Similarly, if we can interpret E, we can say like this, that E sample has the lowest moisture content among all the other samples, and it is statistically significant as compared or with respect to the sample D, but it is statistically similar with respect to the C, B and A samples. Why? Because the same case is occurring as the groups of means of this E sample and the groups of means of the C, B and A samples, they are lying in the same group. So that's why they are statistically not significant. So from the statistical interpretation, we can actually identify and pinpoint the interrelationships among the different samples. That is how the samples vary, how the samples are related to each other, how the samples are correlated, how their samples are linked with respect to one another, and you can also correlate your data with other experiments as well, and also to test your accuracy and to validate that whether your data is really right or wrong. We have to perform the statistical analysis. So with this we have done with the generation of the graph of the percentage moisture content with respect to the different samples, and also we have done the statistical analysis of the interpretation and how can we understand the relationships with the different samples. Now what we will do, in order to export this graph, we will simply go to file, we will go to export graphs, we will go to open dialog, and then a dialog box will come basically where we can select any kind of format that you really want to be exported. So we can have a TIFF file or we can have a JPG file in whatever format you want. Okay, and then we can select in any folder that you want. Let me select the desktop. Okay, so desktop is already there, or we can select any folder that you want. Okay, and then we will click on OK. So it will be saved as graph1 as saying, or you can change the name also as per the convenience. So your graph will be saved in the JPG format, and then you can use the graph in any paper or any article or any thesis that you want. So let's go to the graph. So this is your graph basically, as you can see from here, that the graph of the moisture content along with the statistical interpretation of the samples will come into action. So this is the graph. So you can basically bolden this, as I've already showed in the video, that as we have made the things bold, so in order to beautify your graph, you can also basically you can bold these particular letters in order to show more beauty and add more clarity to your graph. In this way, we have understood that how can we plot the data of the percentage moisture content with respect to different samples, and then how can we statistically interpret our data with the help of the SPSS software. So for this we require two softwares. One is our Origin software, the Origin 9.1 version or any version that you can use, and another is your SPSS software. Here we use version 20. You can use any lower version or higher version as per your convenience. Thank you so much for watching this video. Thank you.